So I'm going to talk about the microbiome and targeted therapy in the intensive care unit. I do have an important disclosure in relation to this talk. I'm a lead investigator of a study looking at probiotic use in the ICU, and we've got in-kind support of the probiotics themselves from an industry sponsor that I'm very grateful for. I think the microbiome is incredibly important and interesting, and it's an area that's exploding in the medical literature. If we go back to the start, really one of our most powerful, if not the most powerful invention we've ever created as Homo sapiens is the scientific method. And it is at its most powerful and most startling when it sort of puts us in our place about our place in the world. Starting with the Copernican revolution, we found out that in fact the sun doesn't revolve around us. Then we found out really that our star is just an inconsequential part of an inconsequential galaxy in the universe. And now it gets a lot more personal. It's only been a couple of years since I found out that within me, I've got effectively a 1.5 kilo organ that no one told me about in medical school. If you don't think it's important and you want to think about how it relates to how we think of ourselves as Homo sapiens, consider that for every one cell that I have that I consider mine, within me there are 10 cells that are bacterial in origin. For every one gene of mine, there are 100 bacterial genes, and for every one of me, there's 1,000 different species of bacteria. So the microbiome, which is not just the microbiota, the bacteria, viruses, fungus within us, but also the interactions with us as a host within a specific niche, is incredibly important in probably in many, many ways that we're yet to understand. Whenever I first start reading about a potential therapeutic target and think about ICU, I develop this sort of existential crisis when I think about previous new therapies in the intensive care unit and their potential for improving outcome. So I start reading about the microbiome. It's all over the literature, and then I think, well, like, why now? How come? Is it just because we haven't found anything else? Is it really important? And without digressing too far, I think there probably are a number of reasons why it's really just surfacing now. The first, and I think it's quite interesting, it really relates all the way back to germ theory, which is a victim in some ways of its own success. We identify specific bacteria that are pathogens. And so from very early on, we think of bacteria as pathogenic. If they're not pathogenic, they may be harmless commensals. But we have this idea in our head that they're pathogenic first and harmless second, and that's all there is to them. And there's probably a second, more subtle part of germ theory as well, is that we really think of bacteria effectively as lone gunmen, right? There's a specific bacteria that causes a specific disease and therefore has a specific treatment. And that specific bacteria just arises out of something, but it is on its own. But really, that doesn't make sense. It's intuitively incorrect. If you want to go back to the very start of creation, then us as eukaryotes have been dabbling with prokaryotes since the start. We phagocytosed early bacteria and created mitochondria, and mitochondria damaged by our current antibiotics. And in fact, this idea of the benefit of commensals is really only relatively new to physicians. If you speak to ecologists, they think clearly it's been an issue from the start. So that's a sort of philosophical why now, but there's also more sort of sciencey reasons why the microbiome is exploding, and that relates to culture-independent DNA sequencing. Basically, our ability to determine the range of organisms within that, and specifically 16S RNA amplification and sequencing. Now, it's been around for 30 plus years, but the high throughput methods that we've needed to bring them close to the bedside, we're not quite there yet, but close enough, have really only been around for 10 years or more, not very long. So it's only been in that time that we've been able to appreciate the range of organisms that grow within us. And it's important to understand this because almost universally, the massive now literature involving the microbiome relies on this 16S to describe what the bacteria are. And it's incredibly useful, but it is not perfect. It's unable to identify organisms that are grown in very, very small numbers, but that might still be useful. It doesn't look at viruses and fungi. They may only contribute less than 5%, but that is a limitation. And it also describes the structure of our microbiome, but doesn't tell us specifically about function. We infer that from some of the causal studies that have been done, the interventional studies, and also what we know about the individual bacteria involved. It can look in great detail at phyla, but at the lowest taxonomical levels, it becomes much more blurry. There's almost innumerable functions of the microbiome that have been purported, and I'm not planning to go through them in detail. What is clear is that there is enormous variation between individuals in the composition of microbiome, but actually when you look at it at a higher level, there's probably only a small number of patterns of the composition that are stable, maybe as few as 3. So although we have huge variation, the ultimate function of the microbiome is the same despite this variation, and the patterns are similar. To just give you one example of the function of the microbiome and the complexity, I thought I'd talk about calories because it's so interesting. So there are polysaccharides that we are unable as humans to digest, but the bacteria within us can break those down into short-chain fatty acids. But that is not distributed equally throughout the microbiome, so it depends on the relative composition of our bacteria, the extent to which they're able to break them down. So how you can have exactly the same diet, but how many calories are available to you and are absorbed depends on the composition of your microbiome. Not only that, the short-chain fatty acids themselves stimulate the immune system and feed back within the microbiome itself. So there's this very, very dynamic process that's occurring. Most of what we know about the microbiome comes from animal studies, in particular germ-free models. So when they've done randomized control trials on germ-free mice, so exactly as it sounds, germ-free, and feed them the microbiome, the gut microflora, from either lean or obese mice, then they're fed exactly the same diet. Within 2 weeks, the ones who are fed the fecal microbiota from the obese mice become obese, whereas the ones from the lean mice do not. So this is very powerful interventional data. I guess the area maybe of greatest interest to us in the ICU is these last two, colonization resistance and modulating the immune response. And again, I don't want to go into the sort of mechanisms in great detail, but the idea with colonization resistance is that our own healthy microflora occupy niches physically and in terms of the nutrients they use that prevent colonization by pathogenic bacteria. And then there's innumerable ways in which there's modulation of the immune response through two-way chat. So what happens when our microbiome becomes disturbed? We get this syndrome called dysbiosis, effectively a change from our normal flora, usually with depletion and a smaller number of overall species. And I guess the bottom line for critically ill patients is we don't know yet a lot about this, but there's a lot of powerful data outside of ICU. If you take healthy people and look at what happens to their microbiome when they get a course of antibiotics like Cipro, you get changes within 3 days of starting, and the normal bacteria don't start recurring till at least a week after you stop the antibiotics, and it takes 6 months to about a year to then stabilize the microbial composition, and it doesn't return to how it was before antibiotics. So there are long-term changes too that are really interesting. In some ways, our interventional data to look at the effect of what we do when we intervene and try and prevent dysbiosis in ICU, sort of we put the cart before the horse. We know more about intervening to prevent this than we do about the epidemiology of it. This is the forest plot from the Cochrane about SDD and mortality. So it sort of relies on the same principle. I sort of think of it as the carrot and the stick approach. If you think of SDD as the stick, where you've got colonization of the gut with pathogenic bacteria, then that seems to be, as one of the most tested interventions in ICU, the most effective thing you can do to improve mortality. But maybe we're just kicking the can down the road, right? Most of these studies are cluster trials, and if the antibiotic changes, cause longer-term problems, maybe the mortality difference isn't maintained because the effect of the antibiotics is longer. Or maybe there's other ways to do it that don't involve antibiotics, that aren't going to cause the same concerns around ecological microbial change. As I said, I think we know very little so far about dysbiosis and critical illness itself. In patients who've had stem cell transplants, we know that domination of the GI tract by VRE precedes bloodstream infection. And now there's starting to be small studies looking at the changes in the microbiome in critical illness, but so far they're very small. We do know that there's rapid depletion in the size and diversity. It's decimated in critical illness, both in septic and non-septic patients. And then there's been attempts to look at whether that can predict outcomes. Diversity, things like the ratio of different phyla or gram negatives, and I think ultimately so far there's not enough data to be sure. So if you think of the microbiome as a potential target, as dysbiosis as a potential target, I guess first of all it doesn't have to be very targeted therapy. You can think of nutritional interventions that might alter it, and you can think of antibiotic interventions that might alter it. Probiotics have been around for ages, since the 1950s. They've sort of got this hippie vibe about them. In fact, probiotic was termed as a way to counter antibiotic. Probiotic means for life, antibiotic, against life. So they got this hippie feel. They've been around a long time, but intuitively, I think they make sense. Here's the Cochrane review, published relatively recently, about the effect in randomized controlled trials of probiotics on ventilator-associated pneumonia. And I think that's a reasonable place to start. We're giving something in the gut. We're trying to replenish our sort of lost healthy bugs, and you've got this sort of gut-lung symbiosis thing happening. And it looks like it might be beneficial, but the studies are of low quality. Since then, there's been a couple of other trials that have come to sort of similar conclusions. There's still huge limitations with what we know about the microbiome. It changes in critical illness, sure. Is that association, or might there be some, is it causation? We don't know. I think a really important limitation or potential problem is the variation in the microbiome. So it doesn't just change between individuals, it changes within individuals over time, and it changes depending on where you're looking at it. So different points in your gut, and in fact, even whether you're looking within the lumen or in the mucosa itself. So there's all these different changes that we've got to think about. As I said before, it doesn't take into account most of it, viruses and fungi. And also there's this issue about structure and function. Just because we have this structural outline of the microbiome, what does it actually mean about its function? I mentioned at the start I'm sort of involved in a randomized controlled trial, and we're trying to address some of those issues. We're going to compare Plantarum 299V, just a single strain, high quality probiotic, against placebo. in sick patients in the ICU. And we're going to do it in a blinded fashion and continue that until day 60, irrespective of whether they remain in the ICU, in the hospital, or have gone home. And we're going to couple that with some longitudinal analyses, both of the microbiome and also the functional output of the microbiome, the metabolome. So I guess in summary, I think this is an incredibly interesting and important area that we're only just starting to get to grips with. It's far too early to say whether probiotics or indeed any other intervention can alter outcomes based on the microbiome as a target, but I think it's certainly worth looking at. Please come along to the Gold Coast, and thanks very much for your time.